Check, 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 check. Hello, 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 hello. Can you hear me? Test one, two, test one, two. Can you hear? Can you hear? Can you hear? One, 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 one. Three, three. We're good? Cool. Please take your seats. The event is about to begin.
If you could, everyone take their seats, please. We'll begin in a minute. Okay, let's get started. I'm delighted to welcome and thank the audience here in person for coming to the Texas Alzheimer's Research and Care Consortium symposium, as well as our remote audience joining us from many academic institutions and research organizations across the nation and across the world. Wanted to thank the TARC Partnership for making this possible, and the committee, the organizing committee, for operationalizing this event. Special thanks to the Oscar Fisher Lecture Committee, made up of the Dr. Charles Nemiroff, if you could just stand, Dr. Bob Messing, and Dr. Nick Barbaro. And this is a very important lecture today because it is the 10th Oscar Fisher Lecture, sponsored by the Mulva Clinic for the Neurosciences at the University of Texas at Austin. And wanted to just say a few words about how the series came about. This was due to the incredible generosity and foresight of Dr. Jim Truchard and his vision and for keeping Oscar Fisher and his research alive, his legacy, in perpetuity through this endowed lecture series. So thank you, Dr. Truchard. Everybody give it, thank you. Our 10th Oscar Fisher lecturer is Professor Karen Duff, who will be speaking to us about tangles and unraveling what we don't know about tau. It is really a true pleasure to introduce Dr. Duff. She is the Centre Director of the UK Dementia Research Institute at University College London and Professor Emerita and Special Lecturer at the Department of Pathology at Columbia University Medical Center in New York. Professor Duff explores disease mechanisms and tests therapeutic approaches to Alzheimer's disease, frontotemporal dementia, and other dementias. Her current interests are exploring the mechanisms involved in the spread of pathogenic proteins within the brain, understanding the basis of selective cellular vulnerability and resilience to tauopathy, and developing new mouse models and cell models to understand the earliest stages in tau pathogenesis, as well as the therapeutic implications of these discoveries, perfectly fitting our goal of really attracting lecturers who are great minds in dementia causes and cures. I wanted to just mention that Professor Duff, following her PhD in 1991 at the University of Cambridge, with Sidney Brennan Department in Molecular Genetics, has had a meteoric rise in the field, publishing many extraordinarily influential papers on her discoveries, and receiving the Potemkin Prize in 2006, the highest prize in Alzheimer's research. And most recently, she received the British Neuroscience Association's Award for Outstanding Contributions to Neuroscience in 2020. And she has been elected as a fellowship of the UK Academy of Medical Sciences in 2022. On a personal note, I will just recall as a resident myself in neurology, doing a required year of neuropathology, when our professor of neuropathology doing work in dementia cases, these are autopsies, where we spent countless hours looking at the pathology under the microscope in autopsy series. At the end of all of his instruction, he said, you know, this is a complete waste of time, because what we need to know is the spatiotemporal kinetics of the diffusion of tau in the earliest stages onward, and how that evolves in specific circuits in the brain. And that is exactly the career of Professor Duff. So we're delighted to have her here, and I personally am really excited to hear her talk. I wanted to just remark that we will have question and answers at the end of the series and at the end of her talk. So Professor Duff, please come and tell us about tangles and unraveling what we don't know about tau. Thank you.
Okay, so is this on? Yes, sounds like it is. So thank you very much to Dr. Padavar for that introduction, and thank you to the committee for nominating me and inviting me. And thank you to Dr. Truchard for his insight in having this lecture series and the prize which went before it. So I'm really delighted to be talking at this lecture series, because so much of my work has been inspired or actually provoked by what you see at pathology. And a lot of the questions that I ask are questions that I've had from looking at pathology of Alzheimer's disease and frontal temporal lobe dementia. And I think they should still say unraveling what we still don't know about tau 120 years later, because really we've come a long, long way, but there's still a lot we don't know. So let me get started. So you'll all have seen this. I'm sure everybody starts with this slide. This is Fisher's original drawings of the amyloid plaques, or the drusen, which means geodes, on the left, and the tau tangles on the right. And I'm not going to try and pronounce what they were called in German, but they are remarkably prescient in understanding that there were different morphologies, there were different densities of the stuff that was inside these tangles, and looking at where they were and their relationship to amyloid. So Oskar Fischer, as you know, was pretty much not recognized for his work at the time, and I'm really happy to see that he's being recognized now more and more for his work, his seminal work on understanding the pathology of Alzheimer's disease. So I'm going to start with a little bit of a rundown of tau pathology, what we have observed from pathology series. And really the thing that most intrigued me, so I started my academic life working with John Hardy on APP and amyloid. And around 1996, I decided, okay, there must be more to these tangles that we see in the brain of Alzheimer's disease. And nobody was really looking at them. They were considered secondary pathology, not important necessarily. A few key standout people were working on it, but it was very much an underrepresented part of the field. And so I started making mice. At that time, it was with wild-type tau, because we didn't have mutations that cause FTD. But what drew me to it was that you have this incredible diversity, both in clinical manifestation of diseases that have tau as the basis of them, but also in their pathology, and now we'll see in their structural diversity, et cetera. So my fascination was that you have one molecule, one type of protein, tau, and you can have all of this spectrum of diseases and different manifestations and phenotypes, and that's what drew me to it. And what I'm going to try and focus on in this talk is really what we can understand from looking at different types of tauopathies. And I'll come back to that in a minute. First of all, I'm just going to walk you through what I mean by the diversity of diseases associated with this pathology. So here you have tau pathology in a number of different diseases. You have Alzheimer's disease, and then you have, which is a secondary tauopathy, considering amyloid is the first pathology. And then you have the primary tauopathies. But the brown stain is the tau accumulating, and you can see the morphology is slightly different with the flame-shaped tangles in Alzheimer's disease and more sort of globular inclusions in some of the other FTDs. You also have involvement of glia in the FTDs, not in Alzheimer's disease. So this one protein can do different things in cells. It can form different morphologies. And it's got quite a lot of diversity, which has confused people as to how these diseases are perhaps related. We have diversity in the anatomical distribution. And this was what set off my interest that Dr. Pasva, I'm sorry, alluded to, which is my interest in the spread of tau between regions in the brain. And it was these observations that there was a distribution difference between the different tauopathy diseases that really sort of started my interest in how tau was spreading and why there are different areas initiated in the different diseases, all with the same protein tau. So you can see we've got a number of, we've got different distributions between Alzheimer's disease and the FTD diseases. And we also have some other syndromes and diseases that have tauopathy as part of the phenotype. And here you've got CTE as a result of brain injury, which is another tauopathy. And the tauopathies are growing all the time. I just had a paper in Lancet Neurology last year, which was discussing how tauopathy was a feature of epilepsy, a feature of not only the brain injury, but also post-encephalitic Parkinsonism as a consequence of viral infections. A lot of these different chronic inflammation diseases lead to tauopathy. So there's a lot of interest now in how that perhaps is linking, the immune response is linking to tau pathology. So the tau protein, just wanted to describe how it looks. It obviously doesn't look like this. This is a linear sequence. But it has a couple of domains, of which the important one is here towards the C-terminus, which is where the microtubule binding domains are. So tau is a microtubule binding protein, and it has these repeat areas which will bind to the microtubules. And we'll come into that in the next slide a little bit more. But this is the business end, pretty much. The C-terminus end of tau is what forms the aggregates that you see in Alzheimer's disease or in other tauopathies. So there's a number of, so a lot of the drugs that are being developed are focusing on that region down at the C-terminus. So we have diversity in terms of the splice forms, right? So tau is, in human adult brain, there are 6 isoforms of tau, and they, oops, sorry, pressing the wrong one. I knew I'd do that. And they consist of 3 different isoforms which have 3 repeat domains and 3 isoforms that have 4 repeat domains. And then they have some inserts here at the end terminus. And the purpose of those repeat domains, as I said, is to bind microtubules. And it's thought that those repeat domains are what cause microtubule stability, allowing axonal transport, and the function of tau in the normal situation. Now, any difference in that splice ratio from 1 to 1 of 3 repeat to 4 repeat can lead to disease. And this is really interesting. If you have too much 3 repeat tau, either through a mutation or sporadically, you get Pick's disease. If you have too much 4 repeat tau, you can get one of the primary FTDs, PSP, CBD, etc. If you have Alzheimer's disease, you have an equal ratio of 3 repeat and 4 repeat tau. So this is really interesting how the different isoforms, ratios are different in different diseases, and how this can be a pathological mechanism leading to disease. And this is some of the new stuff that's coming out. Here's Fisher's original drawing of a tangle, and we're now getting this amazing structural diversity of the tau molecule in the different diseases. It's a little bit hard to see, I'm sorry, down here. This is a table of different structures elucidated by cryo-EM. And this is how you would see these in cryo-EM. You can see there are clearly different structural forms which go with either Pick's disease, Alzheimer's disease, or a range of different diseases. So how does this protein take different forms? Why does it take different structural forms? What causes it to take different structural forms? And importantly, does the structural form determine anything to do with the disease pathobiology? Does it determine which cell types accumulate the tau? So lots of questions based on the structural diversity of tau that are as yet unanswered. So just to orientate you with the pathobiology of tau so that you understand sort of some of the slides I'm going to come on to. So as I mentioned, tau here shown in these little orange dots here on the microtubules, usually located in the axon of a healthy neuron, binding the microtubules, stabilizing microtubules. For some reason, it can dissociate from the microtubules, and those reasons are thought to be elevated A-beta in Alzheimer's disease. There are mutations in the MAPT or tau gene that can cause FTD. There's a syndrome called age-related degeneration, or PART, which shows tau pathology building up, but we don't know how pathological that is. And as I mentioned, there's a chronic inflammation pathway, perhaps leading to tauopathy through some mechanism that we don't know as of yet. So what happens in the first instance is tau dissociates from the microtubules, the concentration builds up, and it starts to aggregate. It's usually a soluble, natively disordered protein, not usually forming aggregates and oligomeric, smaller aggregate forms. But in the case of a disease, it comes off the microtubules and starts to form aggregates. Now, we don't know the order of events that this occurs by. So we do know that it redistributes from its normal place in the axon down to the somatodendritic compartment as a very early stage in tau pathogenesis. It accumulates in the cell bodies. It undergoes hyperphosphorylation. It undergoes conformational change to make these tau conformational seeds. And it can then clog up the normal clearance pathways, the proteasome and the autophagy pathways. But we don't know what order this is, and we don't know, these are double-sided arrows, we don't know what comes first and what leads to what. So we're still really in the dark about the ordering of events. And as was mentioned, this is really a very important part of the field. We need to know what these initiating events are, the order of events, and where we can intervene with drugs. So my job has been more recently to try and develop models where we can start to get at that really initial series of events and understand what has toxicity, what is not toxic, and what we should perhaps target with drugs. So I have used the great diversity of mutations in the tau gene to guide me in how to make my models, both mouse models and cell models. And these are the mutation locations in the MAPT tau gene shown here. And you'll immediately see that most of these mutations occur here in the C-terminal end of the protein, which is represented down here. And a lot of them occur around this exon 10 area. And they can cause either structural changes or they can cause splice shifting. A lot of them are missense mutations that are in exons, but some of them are intronic mutations, which occur between exon 10 and 11, and the function of those mutations, which are listed here, their location within the intron, is to destabilize the stem loop and cause splicing in or splicing out of this repeat domain, which is what gives you the 4-repeat form of tau or the 3-repeat form of tau. So there's a lot of tools that you can use by creating mutant variants of the tau and see what happens when you express this in a mice or in cell cultures. So I'm going to talk today about 2 mutations primarily. This is the mutation at P301S and a mutation at S305N. I also am going to be talking about mice lines that have this mutation here in the intron, but really I'm comparing the effects of these 2 mutations. So we have we have been using mouse models that have been derived from a mouse line created by Takao Macedo at Riken Institute in Japan by his postdoc Naota Watamura, who's now in London with me. And Takaomi's lab had created a mouse where they had switched out the mouse gene, MAPT gene, and put in the human MAPT gene. So they've humanized the mice. They've then introduced, by base editing, mutations into a whole slew of different sites, and we're now working our way through looking at those mice to see what those mutations do. The two I'm going to be talking about are the S305N site with the 10+3, and this is considered to be a splice shifting mutation. Although it's exonic, it's a missense mutation, its primary role is to destabilize splicing, so you get more of this 4-repeat tau formed. The other mutation, P301S, is located within exon 10, and it's thought to cause more aggregation. And you can see in this picture that perhaps we'll get more of these little abnormally conformed structures of tau forming. So I'm going to be showing you data from comparing these two mice. And the goal here was to look at how was tauopathy initiated in these mice. They're an excellent model because they're knock-ins, no overexpression, they're humanized. So these really are sort of first-in-class type mice models to look at really the very earliest stages in tauopathy development. So this is the pathology that develops, and what you're looking at here is, oops, sorry, doing it again. What you're looking at here is this is the wild-type mouse. This is a mouse where you've just changed, put the wild-type human sequence in. And then you look through here and you can start to see pathology developing when you have the S305N mutation put in. This brown stain is AT8 antibody, an antibody against phospho-tau accumulation. And you see it accumulating here, particularly in the CA3 region and somewhat in the hilus. We also get accumulation of tau in the frontal cortex and entorhinal cortex, and as I mentioned, in CA3 and in the dentate gyrus. So this is quite nice pathology. However, we have to wait quite long. This is 18 months old, but that's nothing. I'll show you some even older mice as we go through. So because we always want ground truth, we have looked at some of the tissue from a human with this mutation, and the pathology doesn't look dissimilar. It's not, for example, flame-shaped tangles. It looks more like these sort of pick-like inclusions. What's of interest to us is the distribution, and I have a very big interest in the involvement of the entorhinal cortex and hippocampus, and I'll show you a little bit later. It's one of the areas that's involved very, very early on in Alzheimer's disease, and also in some of the FTDs. It's not usually recognized as an initiating site, but some of the FTDs do have a strong involvement of the hippocampal formation. And sure enough, in our mice, we get involvement of these areas, the entorhinal cortex shown here, and the subiculum. CA3 is highly affected. This is a heat map of tau pathology in this mouse, and these areas that are involved in the hippocampal formation. So we think that this models the humans quite well, and it leads us on to some studies that we're doing, really thinking of these as models for Alzheimer's tauopathy, as well as for FTD. One of the things we're particularly interested in, and this came again from the pathology studies, is why does tau pathology only seem to affect certain neurons? And this is the basis of this idea of selective cellular vulnerability, which is an absolutely fascinating concept. Why is one neuron affected when its neighboring neuron is really completely unaffected, even though it looks very similar? And this has really intrigued neuropathologists and now molecular biologists for decades, even centuries. A lot has been written about the types of cells that are impacted and the order at which the pathology develops and the region that affects. And you can imagine that the type of cell that's impacted will actually lead you to the manifestation of the disease later on, because if you're impacting the entorhinal cortex or the pyramidal neurons in the CA1, you're going to get memory disorders. If you're impacting, say, the frontal cortex, you might get social problems or different types of manifestation of the disease. So this idea of why certain neurons are impacted and when is really an important part of the disease process, and something that a lot of people are putting a lot of effort into understanding. So one of the things that's really held true and is very interesting about disease is that actually these tangles or tau pathology impacts primarily the excitatory neurons. And we can see this. We did a little study in Alzheimer's disease using markers of excitatory neurons. I think this is SATB2, and inhibitory neurons, parvalbumin, and coupling those markers with a tau marker. And here you can see tau in green, SATB2 in red. And you really see a very good co-localization of tau pathology in human AD with excitatory neurons and exclusion from inhibitory neurons. We've looked in our mouse because we want to make sure that things look the same. And here's SATB2 in red with tau in green. And you can see co-localization again, primarily in the excitatory neurons and sparing of the inhibitory neurons. We've looked in FTD cases from human, and again, this holds true. There's something exquisitely sensitive about excitatory neurons to tau pathology development. Or there's something protective about inhibitory neurons that protect against tau pathology development. So one of the things we're focusing on at the moment is a very sort of detailed project to do cell typing. And I'm not going to talk about this very much. It's just starting in the lab. But there are several methods for looking at the cell type of neurons. And we've chosen to use a method called, this is copperfish, which was described by Matthias Mann and was followed up by Kenneth Harris, who is our neighbor in the labs at UCL, using this in situ sequencing method to identify cell types. And the reason that this is a really good method for what we want to do is because you're not only going to identify the cell types through using probes, and they now have something like 350 different probes for different neuronal subclasses, but you can link this then with immunofluorescence. You can do 7 rounds of immunofluorescence, and using cell segmentation, you can put together your signal, your immunolabel, for example, to tau. We're also looking for P62 to identify neurons that have deficits in proteostasis. And so you can now couple immunofluorescence with cell labeling at the single cell level. So this is a really powerful technique for, oops, I don't want to give that one away yet. This is a really powerful technique for looking at the individual neurons types that are affected. And here you can see sort of the granularity that you get. This is focusing on the hippocampus. You can you find a single patch here, and when you sort of go down in granularity, this is a single neuron labeled with various probes, which are bar-coded and color-coded in different colors, and through imaging, you can identify the cell type, and they're called T-types, transcription types, when you define neurons by different transcriptional markers. So this is a method for cell typing that's compatible with the question we have. And the question we have is what cell type accumulates tau? Both in mice, we're doing it in mice at the moment, but this is also compatible with human if you have good tissue. And this is really a powerful method. It's not being used so much, and the reason I'm focusing on it is because it's a lesser known method, because it has considerable imaging-based methods of cell typing are quite tricky. It's all in the segmentation and the alignment between rounds of imaging and washing and etc. So this is, but we're hoping that we'll come out of this really being able to identify not just that it's an excitatory neuron, but that actually it's exact cell type. And that, of course, is going to be interesting because if you look between different diseases, does tau always impact the same types of neurons? And then, of course, you can think about what's the physiology of that neuron. Why is it so vulnerable to tauopathy, or why is it protected if it's the neighboring one that looks very similar? And you can really dial down and try and understand some mechanistic basis of why some cells develop tauopathy and others don't. And this can be used for any disease. This selective cellular vulnerability is common to pretty much all the neurodegenerative diseases. It's particularly interesting for tau because, as I said, you've got one protein and you've got different manifestations of the disease, different areas affected, and we have an active program trying to find what is the basis for this. Is it the structure of the tau? So trying to couple this with more structural studies to look at why tau will impact some neurons and whether the structure of the tau will change which neuron it impacts. So that's a little bit about some of that work that we're doing. And I want to now switch to the other mouse model that we're looking at, and this is the P301S mouse model. So this mouse has the P301S mutation, which is within exon 10. Again, it's a knock-in. But this mouse actually has a very different phenotype. So we have to wait till about 30 months of age to start to see robust tauopathy. By 22 months of age, you might be lucky to see a sparse tangle, but they look very different to what you'd see in the S305N mouse. They're really very similar. They've got the same background, they've got the same tau that this has been inserted to. It's all at endogenous levels. The only thing that's different is you've got a mutation. And we find this really fascinating. I think you're going to be able to see how these mutations are causing some of the pathobiology that goes to giving you different phenotypic expression. So for example, pathology developing much later, the pathology looks much different in these mice. So one of the things we're very interested in is the phosphorylation, the impact of phosphorylation on the tauopathy. This has come and gone in the field, the impact of post-translational modifications, phosphorylation, acetylation, ubiquitination, for example. But we're particularly interested in the phosphorylation. And one of the results that we got when we did mass phosphoproteomics and total proteomics, here's the S305N mouse, here's the P301S mouse, and here's the wild-type mouse. And one of the things we're seeing is this really robust difference between the phosphoepitopes that are in these mice. So here, the MAPT is the tau phosphoepitopes, and you can see by this yellow color, which shows that these phosphoepitopes are very highly represented in this mouse line compared to the P301S. And this is really interesting because we don't see these phosphorylation changes in transgenic mice or in humans, probably because they've gone to a later stage. We're at a very early stage in the pathology here. What we found particularly interesting was that those phosphoepitopes line up here in one domain. This is the proline-rich domain. And this has been implicated as being a domain that's involved in very early pathogenesis. Before you get acetylation, ubiquitination, you get phosphorylation in this domain. And this is also potentially involved in the formation of LLPS condensates, which I think Peter Hislop was here last, perhaps talking about tau condensate formation in Alzheimer's disease. So we're very interested in what this perhaps says about how tauopathy is developing and why they're all focused on that region, and of course why they're not in this other mouse model. Seeding is a very important part of the process, and Mark Diamond has talked a couple of years about this probably. But this is everybody wants to see an aggregate forming and fibrils forming of tau in these mice. Now, we've done a study with Mark to some extent using his seeding assay, and here's the seeding assay where you put lysate from your mouse into the biosensor cell line, and by FRET you can identify whether the tau has seeded onto a fluorescent and formed a fluorescent signal that can be picked up by FRET. And what we see is that only the P301S mice have tau seeds. Here's the S305N, here's the control, the PS19 mouse, and we really only see seeds forming. What's interesting is we see these seeds as early as 9 months. And you remember in that mouse, we don't see much pathology until 24 months, 30 months. So this seeding event really precedes by a considerable time what you see happening in the pathology. And this is a really nice, this is a money shot picture. This is immuno-EM showing, these are gold-labeled tau antibodies, labeling these little filaments that we were able to pick up in the extract from the P301S mouse. We don't see these in the S305 or the wild type. So we really believe these are valid tau aggregates. And if you've followed sort of the tau literature with mice models, you're probably saying, well, who cares, so what? Okay, we've seen a lot of, aggregate forming mice. But these are knock-ins. These are endogenous level. There's no overexpression. This is really unusual to be able to see tau aggregate formation in these sort of mouse models. Going back to the 305N mice, and we haven't done the P301S yet, but going back to the 305N, you remember they had hyperphosphorylation but no seeds, but they actually have degeneration. This is a silver stain. This is a cupric silver silver stain showing these little neurites which are degenerating in the 305N mice. So even though they don't have aggregates, they have degeneration. They also have synapse loss. So we do a study where we can look at the pre- and post-synaptic compartments, and we can count how many are forming an active synapse. And when you look at the 305N mice in different regions, you see a decrease in the amount of synapses formed in this model. And this is not that late. It's around 15 months of age that we can pick this up. So again, they have hyperphosphorylation, but no seeds. But they have neurite degeneration. They have synapse loss. Do they have cognitive impairment? So I'm not showing the slides using the regular battery of tests that we do, novel object, etc. They do have deficits. But I thought I would focus on this. I know I met with some of the women in neuroscience, and several of them are in the psychology department. And I thought I would introduce some new tests of behavior that we're doing that gets at some of the aspects of the disease that isn't captured in most of the memory-based mouse tests. So, for example, this test is the IntelliCage test of social and emotional behavior, which captures symptoms like apathy, perseveration, depression, etc. They look like they hang out at the bar. So you put 16 mice in a cage, and you give them sucrose and things, so you can make them go to different corners, and they hang out and do their thing whilst you're monitoring them. And this will tell you if a mouse is depressed. Obviously, it's not going to go to the bar. So it's a test of some of the aspects that could capture, we wanted to capture of relevance to FTD. And when we do this for the mouse model, I put this in small text just because you can probably appreciate that the S305N are deficient in this test. So we think that these mice show apathy and perseveration problems, which is relevant again to the disease that we're trying to model. So we've been thinking now that perhaps there are two mechanisms by which tauopathy can develop, tau pathogenesis can develop. And this is pretty novel in the field. No one's identified these very early stages in the disease and been able to tease apart whether all tau goes through the same process. We're seeing this split between the S305 4-repeat shifting mutations, which go to hyperphosphorylated tau, versus the P301S, which don't go through hyperphosphorylated tau, but go through conformational change and seed formation. Now, we know from the S305, they also go on to axonal degeneration, synapse loss, and cognitive impairment. We don't know at this stage whether the P301S do, but certainly at the younger age that we've been looking at, they don't seem to show much degeneration or cognitive impairment. So we're wondering if there are two divergent mechanisms, at least for these mutations, and perhaps this is impactful on how we're going to treat these diseases. For example, if you've got hyperphosphorylated tau, but no seeds, you don't want to be feeding these people a seed aggregation inhibitor. And vice versa, if you've got conformational change but with no hyperphosphorylated tau, perhaps you don't want to be giving them a kinase inhibitor. So it might be impactful. However, we've got to, of course, be aware that end-stage disease for humans always ends up looking pretty much the same. You've got seeds, you've got hyperphosphorylated tau, you've got pathology. So at some point, these two mechanisms will converge and give you what you see at end stage in humans. But we're really interested in whether you can use biomarker studies to perhaps pick up what pathway your tauopathy is going through. And sure enough, we've seen phospho-tau in these mice in the plasma, and we've not seen phospho-tau in these mice. So perhaps this mechanism can be picked up in humans in the plasma if you know which type of route of pathogenesis they're going through. Give me a minute to have some water. So that's it for the mouse models. And I really wanted to sort of give you the take-home message that this is not a normal mouse line. These are really going to be insightful for new ideas about tauopathy, which are translatable to Alzheimer's disease, of course. We actually think that the route for Alzheimer's disease tauopathy is probably going through the 4-repeat S305N type of line, because you get the hyperphosphorylated tau. And so that might be impactful for understanding tauopathy in human AD as well. So I wanted to just end up talking a little bit about how the knowledge of where the brain the tangles form in the brain can be used to diagnose Alzheimer's earlier. So I'm going to switch tangents here, but it uses the same mouse model, the 305N mouse model, because we know it has some cognitive impairment. So this study came about from, again, pathology studies. And here you see the staging of tau in Alzheimer's disease, with this starting in the lateral entorhinal cortex, which is known as a transentorhinal cortex in humans, moving medially into the medial entorhinal cortex, and then into the hippocampus and out into the neocortex. So we know that this occurs in the earliest stages of Alzheimer's disease, and we also know that it's prodromal. So this does not relate to clinical symptoms. So we're really interested in using this idea of where tau pathology first develops and spreads to in the prodromal phase to try and identify cognitive tests that can pick up the disease in what's currently the prodromal phase, so before they have clinical symptoms, which of course would allow you to administer drugs earlier. So this area is very interesting. It's full of these cell types that are involved in spatial memory and spatial navigation. You have in the hippocampus, you have the place cells, which were identified by John O'Keefe, for which he won the Nobel Prize. You have in the medial entorhinal cortex, you have the grid cells, for which the Moses won the Nobel Prize. And then you have a whole load of other cells that are involved, which are really interesting. Direction and border cells, so you know which way you're going, how to integrate this information, how to recognize different aspects of the landscape, etc. And what I find really interesting is that these same regions are the most affected in early Alzheimer's disease. As we said, it starts in the lateral entorhinal cortex and moves to the medial, which is where the grid cells are, hippocampus, which is where the place cells are. So we know that it's affecting early AD. And as anybody who's worked with AD patients knows, one of the very earliest features of the disease is that they get lost quite easily. They become disorientated in space and they get lost and start wandering. So we're trying to use this information of the location where the pathology is in the earliest stage of the disease to devise some cognitive tests that might pick up this dysfunction very early. I work with Dennis Chan and Neil Burgess at UCL, and they've devised this virtual reality-based approach to looking at an aspect of spatial navigation called path integration. And they've been developing this test. What it's based on is you have your virtual reality landscape, which is shown sort of like this, and a cone pops up, and you walk towards it in virtual reality. Then it goes down. A second one comes up. You walk towards that. You orientate yourself. That goes down. A third one comes up. You walk towards that. Then you've got to get back to the first one with no cues other than what's in the environment being projected on the virtual reality. And deficits in path integration show themselves on this test. It's been shown that this is quite an early sensitive marker in Alzheimer's disease. And so we were interested in whether we can apply this test also to our mice. So Julia Krupik, who's now at UCL, and John O'Keefe at UCL have devised this mouse-based test. And I'm sorry I don't have the video of it, but here's your mouse. And mice can do virtual reality, which is probably a surprise to most people. But they can, if you run this is the virtual reality that they're looking at, and they have a reward where they can lick a sucrose solution. And you can tell that they can tell from their virtual reality landscape when that lick should be coming up, and they slow down. They're on a ball, they have a headset, and you can tell what they're thinking about the reward when it's coming up. And this is also a test of path integration. It's a correlate of human path integration. So we're able to now do this test in mice, and sure enough, our SCF5N mice are showing a deficit in path integration, almost certainly because they have an entorhinal cortex tau. So I wanted to just introduce you to a new set of studies we're doing, and we're doing this with this Colombian kindred, which I don't know if you're aware of, but this is this huge kindred of presenilin-1 early Alzheimer's patients located in the Antioquia region of Colombia. And this has been worked up for several years. They have Alzheimer's caused by a mutation in the presenilin-1 gene, which is the E280A. And there's about 6,000 family members in this region, of which about 1,000 are thought to carry the mutation. The mutation was introduced in the 17th century thanks to a Spanish conquistador. The average age of mild dementia is 45 years of age, and they have an average age of dementia of around 49, so it's a very fast progressing and also very predictable disease. And so you have all these, this is being worked up by Francisco Lapera and his team, both in the States and in Colombia. And we're very interested in whether we can apply this virtual reality-based test that's been shown in a small number of humans to this kindred, right? Can we identify in the prodromal phase, because you know that people with the presenilin mutation are going to get the disease, you know pretty much when they're going to get, almost to the year of their age, when they're going to get the disease. So can we start to look 5 years earlier or whatever and just keep looking at them and check whether this virtual reality-based path integration method will actually pick up the disease in the prodromal phase? What's particularly interesting about this kindred is that they actually show resilience. Some of the people in this kindred who have the presenilin mutation are spared the disease by 20 or 30 years. And they've been an absolute goldmine to look at genes that are actually making them resilient to the disease. And this is a whole other area of interest for me, is actually looking at not what causes a disease, but what causes you not to get the disease. And I think this is something we should think much more about, is resilience, rather than what causes the disease, but how can we protect the brain using its own sort of defense mechanisms and upregulate those perhaps to prevent disease, succumbing to a disease. And what's really interesting about these is that they actually show tauopathy and amyloid in the brain, that the people who've been identified with resilience. But in this case, and this is weak because it's only an N of 1 or 2 people, it was published in Nature Medicine, but it's a small number. This group actually shows no tau in the entorhinal cortex. It's fascinating. They've got tau in other regions of the brain and amyloid, but they have no tau in the entorhinal cortex. And the people who have the resilience variant, it's in the relin gene, which is in the lateral entorhinal cortex, and they are protected. There's a brother and sister. They're protected for 20 or 30 years from getting Alzheimer's disease. And we think that it sort of tells you that tauopathy in the entorhinal cortex is really key to giving you the cognitive symptoms. Fascinating study, and I just bring it up. There's a lot of potential problems, that it's underpowered, but I really wanted people to be aware that these are the sort of studies that are being done. So coming back to what we're doing, we're using this predictable cohort, and perhaps if we can get some of these resilience, people with resilience, to test out whether this cognitive test is working. We're also not only using the virtual reality, but we're also using an app-based system. And we recently went, here's the team, John O'Keefe, Julia Krupik, Dennis Chan, and myself in my VR goggles. We went over to Colombia, to Antioquia, the University of Antioquia, to see whether this method would actually work in both the university setting, where they bring these kindred patients down to, but also in the field, if they can actually be used, both, especially the phone-based app. So this is us trialing it, and now that they're doing a clinical trial with this VR setup on this kindred in Antioquia. And this is us going into the jungle to see, not the jungle, but going up into the Andes to see some of the cohort in the field. So a lot of them are living in villages. This is Angostura we went to in the Colombian mountains. And we went in the helicopter, and here's Professor Lepera. And you can see we were protected by military personnel because it's not the safest area still. You can imagine Colombia is not very, and they actually had 45 military personnel in the air, embedded in the area for a week before we went, just to make sure nothing happened. You can imagine we've got a Nobel Prize winner with us, John O'Keefe, and 2 women, and poor old Dennis, who probably isn't worth much on the ransom scale, but so this was a real adventure. And just to finish off, this was us interviewing the family members. This woman actually has Alzheimer's disease. You can see how young she is. But this is the family, and we were discussing what's the first symptoms you notice. How do you deal with it? And we got a lay of the land. This is them in their beautifully colored homes up in the mountains, and you really get a feel for how this type of diagnostic would work in the field, actually, for people. So this is taking these approaches out of the clinical environment, which is really important, because I think we're not, to solve Alzheimer's disease globally, you can't be bringing people in for 2 weekly infusions of an antibody and, multiple MRI scans. This isn't going to cut it. We have to get better at diagnosing them really early and then get drugs into them that can be administered out in these remote areas. So that's the end of it. I just thought I'd end on this note of how, these ideas about the pathology distribution can lead right to sort of trying to use that information for more targeted and informed diagnostic tests. And this is my lab. This is beautiful London, just in case you haven't been. This is beautiful London. And here's my lab at UCL. And the two people who are mainly involved in the mouse work, Martha and Naoto, and the two people involved in the cell typing. And of course, I have a whole slew of people to thank, including Mark, but people who've also given me done the phosphoproteomics. And I really want to say a big thank you to all the donors, neuropathologists, and tissue banks that have generated more than 120 years worth of questions for us to try and answer. So thank you to everybody for your attention. Thank you.
Well, thank you, Professor Duff. I'm going to make a few comments, but you will all see that it's an ulterior motive. I want to give Professor Duff a little time to rest her vocal cords.
Yeah, sorry.
Well, first of all, this is just wonderful. I just want to comment that we call this the Oscar Fisher Lecture Series because we view it as almost like a course of cutting-edge breakthrough discoveries that can shape the future of how we view Alzheimer's disease, both causes and cures. And I'm really pleased that you referred to some of the previous lectures. And I just wanted to comment that actually the nomination process itself is to ask the previous lecturers what areas of this breakthrough concept is missing in the lecture series. And Professor Duff received the most votes. So it really fills a gap that we had in the lecture series. So I wanted to, and it's also wonderful to hear that the neuropathology that I was told was a waste of time is now being used to very good use with your work. I wanted to start with encouraging folks to come up to the two mics for questions. And Bridget there is pointing to them. And we do have time for some questions. I'll go ahead and start the first. You mentioned previous lectures. We had a lecture from on the importance of sleep, slow wave sleep, particularly delta sleep, but other staging in modulating. It was really focused on amyloid, and I know from your work you've viewed amyloid in this pathway of potentiation of the tau pathways. So I'd really be curious how you might link these two systems, the sleep circuits and the excitability that you commented on, and how that relates to your work.
Yeah, so one of the sleep centers is the locus coeruleus, and that does have tau pathology in early AD. In fact, Brack had to recalibrate his scale from the first stage being entorhinal cortex to actually 1A was in the locus coeruleus. So there's an involvement for sure of tauopathy there. Whether it's pathological, we don't know, right? So you can see tau there in very young people, but it may not actually lead to pathological expression. But the aspect, so we don't know whether tau pathology or amyloid is causing those sleep deficits in AD patients, and I think a lot of work's been done now in FTD to also see whether that occurs. But there may not be tau in the sleep centers, at least in the earlier stages of FTD. I know several people who are recording from those sleep areas and doing overnight studies. One of the things that Julia Krupik is doing, other than developing apps, is she's got smart cages where you can leave your mice and monitor them over 24 hours for a year and capture all of the dynamics, the sleep dynamics and things. So there's very good ways now of testing how sleep is affected as the progression of the disease occurs. So I don't personally work on it, but some of the mice models are very good. The nice thing with them is they have the endogenous distribution of tau, and we cross them with amyloid-forming mice. And they have the endogenous distribution of APP. So now you're really capturing what is relevant for humans because they don't overexpress and they develop the pathology in the right areas. So that would be great to apply some of those sleep studies to some of these mouse models.
With that, just a quick follow-up clarification. So in that sense, the delta sleep mechanism, the physiological mechanism, could promote not only clearance of amyloid, but actually halting or slowing the progression of the
Yeah.
pathway. Is that a theoretical possibility?
Yeah, it's interesting. It's cause or effect, right? Does the disease cause problems with sleep because it's impacting the regions involved, or does the sleep deprivation come first and cause the pathology? So those cause and effect questions are definitely lookable at in these sort of newer mouse models. And a lot of people are doing that, recording from the brain to see what's happening as well, looking at which waves.
Terrific. Well, I'm going to turn it over to the audience and give you your water bottle back.
Thank you.
Yeah, so we have a question over here, and I can't.
I know, we
Okay,
can't.
so we'll start with the mic on this side. Go ahead.
Hi, I'm Sarah Hopp at UT Health in San Antonio, and really, really interesting work with these new knock-in mouse models and the differences between the phosphorylation and the fibril formation. But I'm really curious, have you looked at the microglia response to these different types of tau, these different types of pathology that's forming, or any other glial responses that are different?
Yeah, I'm weak on glia because it's not my area of expertise. There's nothing overt. I would say there's nothing overt in these, in the 305. It's more in the P301S, but it tends to be when the pathology forms later on. So I don't think there's a big glial response in these mice, and we're not seeing pathology developing in astrocytes, tufted, the involvement of astrocytic tau that you would see in some of these FTD diseases. So no, it might be missing a little bit from this. They may be just too early stage. I would be, we are now looking at much later. We're doing a lot of RNA-seq and proteomics to see if we can capture glial signatures. There may be an oligodendrocyte signal, actually, which has been proposed to be particularly vulnerable oligodendrocytes to tau pathology. But no, it's open for looking at in detail.
Great, thank you.
We have time for a couple more questions. Yes, go ahead.
Hi, Dr. Duff. This is Srinath Kadamagudi from UTMB. Thank you very much for a very interesting talk. So I have to bend down. That was actually going to be my question, which you asked, so I'm going to ask another one. As a pioneer in the field of tau spreading and vulnerability and whatnot, what are your thoughts on whether you think tau is spreading in a spatiotemporal form, in like cell to cell, or whether it may be just regional vulnerability in terms of that it's not able to clear the tau, and it's kind of happening simultaneously? It's not really a spreading sort of phenomenon.
Yeah, it's this. I mean, we did some work where we placed the tau pathology in the entorhinal cortex and then watched what happened later on. And we're able to show that at least in those models where you've got overexpression in the entorhinal cortex, it does spread, and it seems to spread transsynaptically to regions that aren't expressing the tau, and so it builds up. So it is a proper spread phenomena. The problem is validating that in human, right? It's very difficult to know whether this is replicating in humans. There's some evidence from surgical cases where a glioma caused the cut in the cortex, and that patient then went on to develop Alzheimer's disease. They had amyloid in the bit that was separated off, just hanging by a thread from the rest of the brain, but no tau, which suggests that actually the progression of the tauopathy in humans also requires contact or some kind of intact system to occur. But we can't use these mice for those sort of studies because, it could be temporal, it could be actual spreading. But it's a very interesting question. We have other models for doing that. We also have cell models which replicate very nicely what we see in some of the cell mouse overexpressing models, that tau is taken up and secreted.
I have a poster looking at how tau interacts with human synapses, if you'd like to check it out.
Thank you. We have time for one more last question.
Oh, that would be me. Hi. I'm Linda Noble. I'm a professor in the departments of neurology and psychology, and we study traumatic brain injuries. And what strikes me about this is how quickly the brain degenerates after an injury, independent of the severity of the injury, how quickly we can see axonal degeneration. So my question for you is, do you have thoughts about the kinetics of degeneration that you see and what might be driving that?
Yeah, I mean, we haven't done a big kinetic study, right? So we only look at 15 months, so we haven't looked at earlier than that. But the kinetics are pretty fast. So the degeneration comes up, when the pathology is pretty mild and without seeds, at least monitored by Kuprick Silver and the synapse loss. So if you consider that degeneration, it's quite early on. And I think it's loss of tau function. Whether that's the same in CTE, of course, CTE is a very unusual, distribution of the tau. tends to take a long time for the pathology to develop, I understand, years after the sort of original insult potentially, but the degeneration can occur faster. Is that what you're getting? Yeah.
Yeah.
So we're trying to separate out degeneration, synapse loss, cognitive impairment, tau pathology, seed formation, hyperphosphorylation, and that's why these mice are so nice, because we can actually start to place those events. It would be quite interesting to actually do CTE to see if they'll develop CTE. I personally don't do any of those concussive types of experiments, but I think they'd be a good model to look at it because they are endogenous levels. It's been hard to do in the mice models, I gather, these concussive injuries, but.
Well,
Does
thank you.
that help answer the question?
Yes, it does. Thank you very much. That
Thank
was
you,
a
Linda.
wonderful talk.
Thanks.
Well, I just wanted to close by first thanking Professor Duff for a wonderful lecture. Thank you, Professor Duff.
Thank you.
And also thank the audience, both in person and remote, for really disseminating and participating in this lecture series and Professor Duff's lecture today. Also, just to make a pitch that this is an opportunity for Texas to link with these many research institutions, including University College London, exchange programs for our trainees, and you mentioned the Women in Neurosciences. There's just so many opportunities for really spreading great ideas around both to Texas and from Texas out. So for trainees. And I would like to then also invite everyone to the next Oscar Fisher lecture in the series. Please hold your calendar for Christopher Walsh from Harvard on April 11th, 2024. So please mark your calendar. And I'd like to also invite everyone here to the dessert reception in the lobby, and particularly to encourage the trainees to interact with Professor Duff and continue the discussions. Thank you very much.
Thank you. Good. Was that suitable? So we'll see if it's Columbia's a bit dysfunctional. Yeah, I mean, it was a push and a pull. Yeah. So finally, the UK got its act together with, like, it's a small country. It needs to group together and have decent funding. People were doing euthanization, so they got that with the DRI. Yeah. And Columbia wasn't doing it, right? They were splitting up all researchers and giving money to this group and that. Tribal behavior. Very tribal. And the next shiny thing would come along, and the donors would go, and so it wasn't really getting. It wasn't using its strengths. It's a fantastic institution. There's no strategic plan and stuff. But, so it was a bit of a push. My primary was in pathology, yes. Sorry. And then the Tau Institute. We can start walking over. Yeah, the light is in the gray. Much phenomenal.